I'll begin this afternoon session with a brief introduction. Welcome to the Canadian Institute of Forestry's National Electronic Lecture Program. My name is Sharon Young, and I will be hosting today's session. Today is Tuesday, March 25th, 2025, and this is the 8th session in the e-lecture series entitled Advances in Operational Forestry, Tree Planting, and Carbon Mitigation Research. This series is a collaborative effort coordinated between the Canadian Forest Service and the Canadian Council of Forest Ministers. The CIF-IFC is very pleased to collaborate and host these webinars. For today's session, we are pleased to have Vishnu Seshan, who will give an exciting presentation titled Imagining Delayed Luminescence and Ultraweak Photon Emission from Plants, a novel approach for nighttime drone forest monitoring. To kick things off, Vishnu is a Ph.D. student in physics and neuroscience at the University of Calgary. Originally from India, he completed his Master of Engineering in Molecular Bioengineering in Imperial College London. His research focuses on quantum biology, and he aims to leverage his interdisciplinary expertise to translate scientific discoveries into impactful healthcare technologies. With that, I will now pass it over to Vishnu.
Thank you, Sharon, for the kind introduction, and good afternoon, everyone. So I'm Vishnu. I'm a third-year PhD student at the University of Calgary, and I'm doing an interdisciplinary PhD in physics and neuroscience. So originally, my colleague, Dr. Vahid Salari, was supposed to give this talk, but unfortunately, he couldn't be here today. So in regard to that, I've been informed to put together a presentation, and it was last minute, so I apologize if that does shine through, but nevertheless, I hope that I'm able to share some useful information that everyone finds interesting and can take something out of. So together, me and Vahid work in a section of the Quantum Cloud Lab, which is supervised by Dr. Daniel Oblak, and this is located within the University of Calgary. So without further ado, I'd like to get straight into my talk. And just before I start with the technical stuff, this is an outline of what I'm presenting. So the first little section would be an introduction as to what we do, how we do it, and why it's exciting for us. And then I'd like to pause for a few minutes for any burning questions about what it is we do. And then I'll proceed into showing you some experimental results that we've had and what that means in the context of our research, as well as point you in some future directions in which our research can be implemented and be useful, especially in the context of monitoring the health of forests. And then after that, finally, I'd like to leave some time at the end to have any questions or feedback from the audience, and I hope that would be an engaging discussion to finish things off. So I hope within the introduction, I'd like to just answer the following questions. So that would be, firstly, what is biological light emission and how do we detect it? Secondly, we're interested in two kinds of light emission. So I would like to also explain what the difference is. And lastly, what do we know about the molecular mechanisms of what we observe? So biological light emission is a broad spectrum of different kinds of light emission. So this can be largely grouped into two categories. There is induced light emission, which requires an external energy source to induce photon emission. This can be in the form of light, it can be in the form of heat, radio, sound, electricity. These are the common categories that people know of today. And otherwise, it could also be spontaneous, which means it doesn't require any kind of excitation and it happens as a sort of continuous process. So this is black body radiation. It's the process by which infrared cameras work. And this happens when mostly in the infrared range. There's bioluminescence, which is a chemical process that happens in certain living beings. And there's ultra-weak photon emission. So this is different from the other two because firstly, this arises due to metabolism. And so far in the literature, we've identified the following sources. So they can be triplet carbonyls, they can be dimolar singular oxygen, or they can be, especially in the case of plants, singular excited pigments, which do include chlorophyll. However, there are many other mechanisms by which this works that are still up for discovery. So we're interested in this range of biological light emission, as well as another phenomenon known as delayed luminescence, which comes under the photoluminescence category. So if you look at it from a time perspective, you see that there are many things occurring at different time scales. So you got the prompt fluorescence and the phosphorescence, which might be more familiar to people, and that happens at pretty short timescales. And then you've got things happening at slightly longer timescales, and in the case of ultra-weak photon emission, this happens continuously. And so this area of at least the temporal scale of emission is interesting to us, mainly because they are more representative of the biochemistry of the living system. And so there are two kinds that we're interested in. There's delayed luminescence, which is a form of induced photoemission, which has a intensity of around 100 to 1,000 photons per centimeter square per second. And this is induced, so we need an external light source. So we can play around with different excitation wavelengths to fully capture the spectral dependence of delayed luminescence as well. And then we have ultra-weak photon emission, which is a spontaneous process, mainly arising from metabolic activity. And this is a much weaker signal, but this is independent of any light, so it happens even in complete darkness. So here we have this is just a camera image of a leaf where some benzocaine gel has been applied, and this has been illuminated under red light. And then to capture the delayed luminescence, we image after about a 30-second delay for 5 minutes. So this is just the intermediate. So if I go back to the start time scale, we arrive around here. And we get this pattern where the regions where the anesthetic has been applied is dimmer than the rest of the leaf. And then we wait for another 15 minutes, 15 to 20 minutes to allow all the delayed luminescence to dissipate. And then we're left with just the continuous photon emission, which is the ultra-weak photon emission. And then we get this pattern here, which is interesting because it shows that the regions where this anesthetic gel or benzocaine has been applied, the regions are brighter than the rest of the leaf. So this is interesting because it suggests that these two phenomena are biochemically different. What makes it interesting is that we almost have two different lenses to view physiological processes in the leaf. One with the delayed luminescence frame and one with the ultra-weak photon emission. So they tell us inherently different things about the plant, and that's why we're interested in two different kinds of light emission here. So the main mechanistic difference comes from the excitation of the excited states. So firstly, we're not exactly sure what the different excited states actually are. That's still an active area of research. But we do know that in the case of delayed luminescence, the excitation comes from an external source of photons. While in the case of ultraweak photon emission, this can be in the form of a chemical energy transfer, which usually happens in the case of metabolism, where a lot of secondary metabolites, which are unstable in nature, form these chemically excited states. And these excited states obviously have a lifetime, and their nature is to decay back to the ground state. And when they do so, they can emit a photon. So that's the inherent mechanism of how these two processes work. And the main difference obviously comes from how the excited state is formed. And then so one thing we know for sure is that there is a common set of chemicals known as reactive oxygen species, which are just highly reactive substances containing oxygen that are usually formed as a byproduct of metabolism. And these tend to react, but they also have very um, useful uses in biology in general, especially in the role of signaling. And what they do is form these electronically excited intermediates, as I've shown before. And these excited intermediates, when they decay, will give rise to either delayed luminescence or UPE. So we can essentially leverage these forms of biological light emission to essentially monitor the activity of reactive oxygen species. And that's really powerful because they come from a wide range of sources. So in plants, the main contributors are photosystem 2. So that's the site of photosynthesis. Researchers do believe that it's photosystem 2 that's responsible for the generation of reactive oxygen species that are relevant to UPE and DL. But photosystem 1 also does generate reactive oxygen species. And then another huge source is NADPH oxidase or NOx. However, reactive oxygen species can also increase from just conditions such as injury, infections, and as a result of enzyme activity, and mainly also from the mitochondria, which is a huge site of oxidative phosphorylation and respiration. So as a result, we can use the light to kind of monitor the levels of reactive oxygen species from various sources and understand sort of the metabolic landscape of the biological system. And then we had an initial hypothesis that was because the relationship between ROS and UP and delayed luminescence is well established, we can leverage that connection to use it as a non-invasive marker of various physiological processes. And this has been done in literature before, but we are a new lab and we do have all this technology that we want to kind of use. So our first goal was to kind of do some experiments where we can differentiate the light emission patterns in stressed leaves versus healthy leaves. And then so we have quantum sensors. So because the intensity of light is really small, so it's several orders of magnitude below the visual threshold. That's why we don't go around seeing glow in the dark people or plants. So we need highly sensitive detectors, and these come in the form of generally quantum sensors, which are used for single photon applications in other areas of research, but they have been repurposed for detecting biophotons and delayed luminescence. So the first detector that I'll talk about is the electron multiplying charge coupled device, or EMCCD camera, which generates a 2D image of the biological sample. So it gives us sort of spatial information of how UPE intensity varies between sort of different regions of the leaf and computes an image. So the advantage of this is that the EMCCD camera is really sensitive. It has about a over 95% quantum efficiency. So it's highly sensitive to these photons. However, the one drawback from this is that you need a decent amount of exposure time in order to generate a high quality image. So whatever spatial resolution you're trying to increase, you have to compromise on the temporal side of things. So if you want to get a good image of a leaf, you can do 30-minute images, or generally, if you want sort of high quality ones, an hour is more appropriate. So that is obviously not great in terms of sort of more temporal applications. So we have another detector that's a lot more simple in that context and a lot cheaper, which is the photomultiplier tube. This doesn't generate a image, but instead generates a one-dimensional time series. So, however, the important thing is that the time resolution can be as small as under a second. So that's where it greatly improves on the camera. So we are trying to find sort of good applications for both these detectors in terms of our research. And then lastly, I'd like to talk about just another kind of detector that we have in the lab, but we're still kind of working on getting it to be able to detect biophotons, and that is the superconducting nanowire single photon detectors. So these are more familiar to people working in quantum physics, as they're usually found in a cryostat at very low temperatures. And usually there is this superconducting chip that is conducting as at a basal state, but when a single photon arrives, it disrupts that conduction to form a resistive period, and this is the operational principle in how the SNSPD detects photons. So this we haven't really been using these for applications just yet, but hopefully this will be a thing in the future for our lab. So this is how the lab looks like. So this massive tent contains a camera. So this is the EMCCD camera where there are water chillers attached to maintain a minus 95-degree temperature on the sensing surface, which makes the dot counts go down and makes this camera really sensitive to light. And then this corner here is the box where our photomultiplier tube is mounted. And so we tend to really isolate our samples from the ambient light just around so that we can maximize the signal that we detect. So this is an example of how a delay luminescence measurement is conducted. So we have the leaf under the camera here. I don't know if that's too obvious, but there's an optical fiber that gives a laser. This in this case is red light, but we do have other colors as well. And then the sample is illuminated for a fixed amount of time, and the light can be turned off from a switch outside the tent. So this makes it useful for sort of conducting experiments without being invasive in terms of entering the chamber and playing around with things. And yeah, it like we can operate from a safe distance and without compromise of our experiments. And then we also have the SNSPDs, which are not shown in a cryostat just next to this. But again, it's still a work in progress for those to be able to be used for biological light emission detection. And then this is the end of the introduction. So hopefully you guys will have learned what ways we're doing and how we do it in terms of the different equipment that we have. So I'd like to pause here for any burning questions, but if not, I can move forward to the end. So I'd just like to ask Sharon if there are any questions right now, and if not, I can just continue.
I haven't seen any questions in the Q&A pod or in the chat yet, so I think we can go ahead.
Okay, perfect. So I will continue with the next part of the talk, which focuses on some experiments that we've done to kind of characterize the effects of, well, delayed luminescence and ultraweak photon emissions on different systems. So firstly, I'll talk about the delayed luminescence and how we can characterize spectral differences as well as the differences when the leaves have a stress applied to it. And then the same, we'll talk about the same when it comes to ultraweak photon emission, as well as a type of analysis known as time series analysis, where we analyze the signals from the photomultiplier tube and use the time series statistics to classify stress in that context. And then lastly, we also do measurements on germinating seeds. So I will show you some of those results and what we're doing in that context. And lastly, to tie this all into sort of a bigger picture goal, I would just like to point out the few applications that we see our research being implemented to. So these are the kind of research directions that we're working towards, and this is kind of the takeaway in terms of how our research can be useful in terms of everyday technology as well. So, yeah, plant delay luminescence usually lasts about 15 to 20 minutes after illumination. So, as you remember, this is our setup for how we do a delay luminescence experiments. And this is just some examples of different plants that we've done. So, this is a cactus, and this is a Venus flytrap, which has images accumulated for different amounts of time. So you can see that we have sort of reliable, at least a reliable camera to capture the delayed luminescence from these species. And we also found that delayed luminescence is influenced by stress. So in the case of a regular leaf, you have sort of a smooth, uh, homogeneous surface, whereas here you can see that at the site of the injury, you don't have as much luminescence as the rest of the leaf. And over here, we have a part of the leaf that's burnt away. And you can clearly see here that that part doesn't emit as much as the rest of the leaf. So you are able to characterize the effects of stress on the leaf, and that's pretty powerful in itself. But we wanted to take it one step further by not only monitoring the spatial differences, but also the temporal. So what we did this time is we took a kinetic imaging framework, which captures images at a regular sequence of time. So we did 1 minute images for the first 15 minutes and found that the trend sort of reversed, as you can see. So in the first minute, you can kind of see that this is the site of the injury and that's dimmer than the rest of the leaf. And this intermediate, you kind of see that this part is starting to brighten up. And this is sort of the final product of that, where the injury site is much brighter than the rest of the leaf. So we found that like the the timing of imaging is also super important in terms of what you observe on there. And well, this makes it this inspired us to sort of combine both approaches and do a spatial temporal approach, and that would be kind of a complete toolbox of analyzing these luminescent signals from leaves. And so we also did the same for a burnt leaf, and here you can see something a bit more interesting where this burnt region sort of fragments into two, where there's a fully burnt region that is extremely dim. but there's also there's an intermediate band of brightness around. And that's pretty exciting because that signals some kind of intermediate zone where there is high level of metabolic activity to mainly, I guess, mediate the damage caused by the burn. And this is exciting because we can monitor sort of the metabolic profile of that intermediate zone and understand the stress response of plants. So overall, this data is pretty interesting to us, and it allows us to kind of identify spatial and temporal signatures of delayed luminescence of different stresses. And one more aspect, one more variable, I guess, of delayed luminescence is also the excitation wavelength. So I showed that we excited the leaf with red light, but we can also do it with other colors. And the delayed luminescence intensity varies between the colors. And what's so interesting about this pattern in particular is that it is largely resemblant to the absorbance of photosystem 2, which is in literature reported to be one of the main sources of delayed luminescence. So, with the shape being similar to that, it's definitely exciting from a research perspective. And so now that this was done on a single leaf, but we want to sort of take this further and build a data set of multispectral delayed luminescence, where this is done in the same 5 colors that we have in our lab. And the data is fitted then to an exponential decay model. And so we have some preliminary data, but that is not being shown today. But we do see that the decay profiles differ in different colors between different stresses between the regular leaves with the burnt leaves and with the injured leaves, and how these leaves vary when they're stressed in different colors is also pretty fascinating. So there's a whole rich data set that we're trying to understand in terms of that. So hopefully we get some information to build a model that sort of determines whether the leaf is healthy or stressed depending on the delayed luminescence decay profiles. And that would be a valuable indicator of plant health. And so that's a step that our next experiments are kind of pushing towards. So this is one part of the research that we do. We then move on to ultraweak photon emission, and as I'd like to call it for today, the spontaneously emitted signals of floral life. So as you can see here, we have different plants. So we have seeds, we have moonbeam seeds that were germinating for 3 days, and you can see that the seeds are bright and we can detect them. Sorry, just a technical difficulty. Um, so yeah, and then we move on to the leaves. So we have these dwarf umbrella tree leaves that they're incredibly resilient. So even after you pluck them from the stem, they last about 2 weeks, and we can get reliable signals from them. So they are what we base most of our imaging on early on. And you can see that there are signatures of injuries. So I had applied a cut, a mechanical cut to every leaf, and you can see that on the image. And what was done in this particular study was that each leaf was treated with a different chemical that altered the UP of the injured site. So I'll explain those results in a few slides. But we also did some preliminary studies on the Venus flytrap. And this is the image that we got from that. this was still kind of early on in our like in the history of our lab, so the image wasn't as great quality as what we get today, but it was still interesting to see that we can get it from different plants. And then so like with the delayed luminescence, UP also has specific spatial signatures. So here with the burn, you can kind of see that really bright intermediate zone. And then there's another intermediate. So this was like 24 hours after the initial stress was applied. So we can kind of see that the zones are sort of transitional, and they can the evolution of this sort of bright area where it sort of widens out is pretty interesting, and we would like to study that further. And then we have a leaf trio with benzocaine, which is a anesthetic. It's a local anesthetic used on dental care and skin. But so they mainly affect sodium channels, but we kind of wanted to see what it does in terms of the UPE signal. And we found that it really enhances the brightness of the injury site. And then the same thing with hydrogen peroxide. However, it also increases the brightness overall. So that's an important signature of stress. And then this is all in comparison to the regular leaf. So I hope you can appreciate that there are also visual differences that one can see straight away. But we wanted to go one step further and also understand the numbers. So what we did was we conducted this experiment where we were interested in this biomechanical injury. So as soon as you cut the leaf and image it, there is that bright spot that flares up every time at the specific to the injury. So we want like this was kind of done by accident where I accidentally sort of spilled a chemical on the injury site and it happened to sort of be more bright. So I wanted to try and see if that was kind of a chemical specific response or was it just like general stress. But it turns out that each chemical has sort of a different signature in terms of the emission profile. So the y-axis is the ratio of the non-injured section versus the injured section. And you can see that especially with the anesthetic, so that's the benzocaine, this UPE just shoots up to about 12, a factor of 12 greater than the rest of the leaf. So this is particularly interesting, especially in the context of anesthetic biology, where for something that kind of affects sodium channels, you wouldn't necessarily straight off the bat imagine that it would play around with the light emission so much. So it's particularly interesting in the context of that, but also sort of secondary functions of anesthetics and especially what they do in a plant injury where the sodium channels obviously play a different role to the ones in humans. So our research will focus on understanding that a bit more and how sort of the chemical landscape sort of reacts to the anesthetic in that sense. And then this is the time evolution, which also suggests that whatever effect we see is present for a extended period of time. So even at 16 hours after, the injury site treated with anesthetic is still so much relatively brighter than the other conditions. And so that sort of piques our interest. And we always kind of want to assess any problem we have on two scales, the spatial and the temporal scale. So you can understand that these two pieces of information sort of tell us different things about the data that we have. And then we move on to the seeds. So we use UPE to monitor the real-time germination of seeds. And usually people have done this with a photomultiplier too, but our work is all the first instances of this being done on the camera where it's in a video format. And let me just play this video here. So before I play the video, I'll just describe to you what's going on here. So there are two dishes of seeds, one treated with H2O2, and one without H2O2. So H2O2 conventionally is a agent that increases ultraweak photon emission, but specific to the context of seeds, low concentrations of H2O2 can also improve the rate of germination. So we kind of we wanted to understand if we can obviously see that effect with the UP. So let me just play this video for you, and I hope you can appreciate the way that the UP spreads out across the dish, and that's pretty interesting to us from not just a biology perspective, but also sort of how each individual seed reacts to each other. And so we're trying to do some interesting stuff with these videos where we convert them to correlation maps, where we map the correlation between two seeds and uh, with regard to the photon emission, where each seed has this sort of shape where they peak. There's a peak metabolism. This is to do with how the seed operates generally, where once we pour water in the imbibition step, the seed coat breaks down, and when it does so, there's a huge spike of metabolic activity, and we believe that's what we see with this peak here. And so we compute sort of the statistics and the dynamics of the peaks, as well as how the time evolution of each seed correlates with each other to understand sort of the emergent properties of so putting 20 seeds into a petri dish and allowing them to germinate, whether we see that the seeds are actively communicating to each other, especially with regard to um, the fitter seeds, the ones that germinate faster, um, signaling the other seeds to germinate faster than they originally would. So it's interesting, and it's all very early days in terms of our findings in the context. So we're trying to just uh, plug together and see what these correlation maps do and see if they give us any indication of how these seeds interact and the difference that different germination conditions make to these correlation maps. So yeah, I'm excited to work on that stuff, and hopefully I can share that with people next time. And lastly, we jump from the images to the time series, and we've come up with a time series analysis framework to identify oxidative stress. So a layman explanation of oxidative stress would be when there is an imbalance of the reactive oxygen species that we saw earlier, which is associated with UPE. So in cases of stressful conditions, the concentration of these chemicals can increase, and when that happens, it generally overwhelms the cell. And so usually the cell has a defense mechanism by another group of chemicals called antioxidants, which react and reduce these highly reactive oxygen species and neutralize them essentially. So oxidative stress occurs when the reactive oxygen species concentration gets so high that it overwhelms the cell's natural antioxidant defense systems. And this is obviously detrimental to the health of the cell. And we thought this would theoretically result in altered UP patterns. And that was sort of the fundamental crux of our analysis. So this was originally thought of from a clinical and diagnostic perspective, where oxidative stress is implicated in the progression and onset of many prevalent diseases. And this comes from humans, so stuff like cancer, neurodegeneration, cardiovascular diseases, respiratory diseases, diabetes. So quite a lot of things where oxidative stress is the early sign, so an early warning sign. And so this can also be thought of in the context of plants where oxidative stress might be sort of a warning sign of certain risk conditions in the forest, such as deforestation or drought, or what can be thought of as potentially a fire risk, as well as sort of widespread disease and infection, as well as another thing, not necessarily to do with stress, is also just the diversity of plants. And oxidative stress could be an indicator that there is a parasite growing in a forest that is negatively affecting a particular species of plant. So there are interesting things that we can do with it in terms of um, applying it to a forest perspective. So how we do this is essentially using the photomultiplier tube. And this is essentially a cheaper alternative and low cost, not as much cooling required. So it's a pretty easy setup. And the motivation for doing this experiment was, well, the camera is really interesting, but it costs a fair bit of money. So how could we implement this in a low cost fashion that can be sort of more accessible to a wide range of people who could benefit from it? So we use the photomultiplier tube, just take UPE signals for about 5 minutes. And this is the form of a one-dimensional time series. And that time series goes through a set of pre-processing where we determine quantities, including the statistics, the entropy, and the nonlinear dynamics of the system. And these are all plugged into a machine learning model that classifies if the leaf is healthy or stressed. And so these are some preliminary results of what we did. And we found that we were able to classify leaves with up to 90% accuracy of whether they're stressed or whether they're healthy. So this is determined by the confusion matrix, where the diagonal elements are correctly identified results and the off-diagonal elements are the incorrect ones. And this element here, where the predictor label is no stress, but it's actually stress, we call this a false positive. And the algorithm is designed to minimize the occurrences of this because it's obviously detrimental to misdiagnose anything that's stressed as healthy and not dealing with the situation and potentially making it worse. So we are trying to minimize those instances. And what's encouraging is that these preliminary results show that this technique could serve some potential in determining the oxidative landscape of a leaf. And then finally, we want to build this to encompass not just plants but also hopefully clinical patients where we can monitor their UPE from using the PMT or a camera, make a classification model similar to this, and then predicting what disease they might have based on the signals, since our hypothesis suggests that the metabolic landscapes of these diseases are different, and we can use that to determine sort of which disease the person might be having. So this is our long-term goal in the diagnosis context. But we also, well, this is the highlight of the talk today, where we can literally mount the detection system on a drone and monitor forests in the night. And the dark counts of a dark sky is, well, it's pretty low without any sort of light pollution. So we still believe that we would be able to get meaningful signal in terms of delayed luminescence from trees. And what we envision is that the drone picks up signals and sends it via Bluetooth to a processor. And the processor could use some machine learning and use some data analysis to determine whether the forest has any sort of problems that people need to be concerned about. So it's not just in terms of health. It could also be liaised with ground level teams where um, once people identify an area that's under risk, the ground level management sort of goes and decides what the best mode of care is in that context. So we imagine that the drone technology would be integrated to forest management and how especially the Canadian forests are already maintained. And well, we hope that our technology can improve those conditions and not have as much devastation as we've seen over the years with regards to forest fires. So yeah, and then lastly, this is kind of an extension of what we can do with the drone. Is that we recognize that each leaf has different delay luminescence characteristics, whether that be the intensity of the signal or how it varies temporally. So we want to. This is just a idea at the moment, and we haven't really done any experiments to kind of confirm that just yet. But we could modify a model where we don't look at if a leaf is healthy, if a plant is healthy or stressed, but we look at sort of the collective signal and with the decay elements being different for each species of plants, we could essentially identify the biodiversity of a particular area of the forest and potentially use that as sort of an indicator of forest health, where if it's home to a wide array of plants, it's obviously a much more richer ecosystem. So yeah, we haven't done any experiments on it, but hopefully in the next year or so we can work towards what that platform might look like. But yeah, so that's the end of the technical topics that I have to present today. So just to conclude, what we do on the research end is generate a toolbox of analysis techniques to extract information about living systems, mainly by sensing their biological light emission. So this could be either delayed luminescence or ultraweak photon emission. And then we also say that the work that we do will complement future ventures in applying this technology to monitor forests. This is the CFI, so we are targeting sort of the applications in a wildlife management perspective. Um, but these include, as I mentioned previously, the diagnosis of diseases, the monitoring of plant health, and monitoring of biodiversity. So that's the end of my talk, and I hope that I've provided some valuable insight for the members of the audience. And I'm happy to answer any further questions. Just before that, this is an acknowledgement of all the collaborators that we work with and the partners who fund our research. But yeah, this is the end, and I'm happy to take any further questions.
Thank you, Vishnu. That was a great presentation. Before moving on to the Q&A session, there is a post-webinar survey for our participants to fill out, and I'm going to launch it now. So there are 2 questions in total. You may need to scroll down to see the next question. And Vishnu, I think right now there are some questions for you to answer them. Are you ready?
Yeah.
Okay, so the first question is, hi Vishnu, thank you for the fascinating talk. I have a pair of questions. The first question is, have you compared some of your DL, UPE signals by direct quantification of metabolites, for example, fluorescein or DAB detection of ROS? And second question is, have you considered using DL, UPE to test analogs or agonists of well-described stress signaling pathways like treatment with plant hormones? like SA, JA, ethylene, and ABA?
Yeah, thank you. Thank you for those two questions. Those are very interesting questions, and they are relevant as well because we are coming at this from a physics perspective. So just the ecosystem that we have is not really built for sort of like large scale biology experiments. So we don't necessarily have the equipment to detect the concentrations of the metabolites just yet, but we are sort of working on getting that system implemented really soon. So we will be excited to um, get that data and obviously correlate um, the molecular um, the molecular results with our signals to ultimately understand the underlying mechanism. So that is something we have thought about, and there are plans to do it in the future. And regarding the second question, in terms of sort of the stress pathways, again, that is also something that can be done in the future, where we still have to get a reliable setup in terms of the on the biochemistry end with all the chemicals in place and things like that. And we just don't have that at the moment. But we are always sort of interested in collaborating with people that might have the expertise on these fronts, and we're always hoping to do better research and uh, over that. But yeah, thank you again for bringing these questions up, and they are sort of things that we have thought about, and hopefully you will see them being put into action sometime soon.
Thank you, Vishnu. Next question for you is, I see the application of this plant study science really beneficial to study the specific impacts of different pathogens and environmental changes. How far away are we from using this process, not only in the lab, but from an overview of forests using the drones? Thank you.
Thank you for that question. It's obviously exciting to kind of determine where we're at in terms of our end goal. And right now, I'd say that it's not really close the timeline in terms of implementing the drones. So I think what's missing right now is obviously doing sort of reliable large scale testing of delayed luminescence and UP characteristics on, well, we've done it in the lab so far. We've done it on just a few species, but we kind of need to do it on a lot more diverse range of wildlife that we find in the forest. So there is a whole classification step of that. And then there is sort of scaling up the model so we can kind of match the complexity of the data, like of the specimens that we would find on the field. And then there comes the technology side where we still have work to do in terms of integrating the quantum sensor on the drone. And especially with the drone, with measuring the signal, we need to also maintain reliable control of the drone's flight pattern. So that would require a lot of research in those areas of research. So it's still kind of a distance away, but I think it's more exciting now that we have the experimental data to show that this can be done, and that now we just require sort of people with different expertises to come together and implement sort of the final parts of that mission, essentially.
Thank you, Vishnu. Next question for you is, are you working directly with forest professionals to collaborate on further studies and application to operational forestry?
So we are not we're not right now, but our that is our goal, and that was our goal to kind of come and present here as well, where we are sort of hoping to liaise with um the forest professionals, the agencies involved in it, to like to collaborate and do some more focused studies to sort of make this dream a reality. So we aren't working with anyone at the moment, but we're always open to liaising with such people. So if anyone happens to know people that we can talk to or reach out to, like, we're always more than welcome to follow up on that.
Thank you, Vishnu. Last question for you is, can we use a drone equipped with an EMCCD camera to analyze your current work? If not, what potential challenges might we face?
So with with the EMCCD camera, there are concerns about mounting that on a drone just because it is quite a heavy payload, especially with implementing a cooling system in place. And because you need images, you'd require the drone to be absolutely still during that imaging session. And this might not necessarily be feasible in terms of an application in this scale. So we might have to move away from a camera and instead implement a photo multiplier tube. But well, like theoretically it can be tested, but the caveats of testing it at this current stage would be that we don't have enough samples, because the plants that we have tested it on, we don't find in the forest right now. So again, we need to go out to the field, collect a few samples, and do our tests on that. But once that's done, I guess like what we need essentially is um, collaboration and some expertise from people who manage the forest in terms of what species might be useful to test and do, like on our side, compute those analysis, develop a toolbox. Suppose we develop it, for example, conifer trees, we can at least test that in the forest. So right now we can't do that with the species of plants that we're working on. But again, this would require sort of us being given some structure by someone who's really familiar in forest plants.
Thank you, Vishnu. I think that's all the questions we have in the chat, in the Q&A pod at this moment. But if anyone has any follow-up questions for Vishnu after this webinar, please feel free to reach out to us and we will help connect you with him. All right, I would like to bring this session to a close if there are no further questions. Thank you all for participating in our CIF electronic lectures. And once again, thank you very much to Vishnu for the great presentation. Bye, everyone.
Yeah, thank you. Thank you for having me here. Goodbye.